Good morning, participants. Myself, Dr. Vidya Hinge, Assistant Professor, Department of Plant Biotechnology, Vilasrav Deshmukh College of Agriculture Biotechnology, Latur. So, I am delivering the seminar on the CRISPR Cas9, a tool for targeted genome editing in plants. For the sake, I have divided my topic into the following content: introduction, genome editing, then CRISPR-Cas9 mechanism of genome editing, then protocol for CRISPR-Cas9 genome editing in plants, then different crop improvements done by using CRISPR-Cas9, biosafety regulations, and finally conclusion. So first of all, introduction. If we see the different genome modification methods used for crop improvement, several breeding techniques are used in earlier phase. Later on, the mutation breeding was found very promising technique, but it is also associated with the several disadvantages like all the mutation breeding techniques that are non-specific, random, and they require large population for the screening. Further, genome modification methods like transgenic methods are very popularly used, but they are also associated with the disadvantages like multiple transgene integration and the vector sequence insertion, and as well as they are associated with the biosafety concern. Further, the microRNA and RNA interference techniques are also used for the crop improvement, but they are again have the disadvantages like the variation in silencing level, they are not stable over generation, and they have the off target effects. But in recently, the genome editing techniques that are very popularly used, and these genome editing techniques have overcome all the disadvantages of these all the genome modification methods. In this genome editing technique, they are very specific, they are precise, faster, and again they are biosafe. So let us see what is the genome editing. So genome editing, it is precise manipulation of the DNA sequence, insertion, deletion, and creation of desired codons. So, if we see this genetic genome editing, it is a genetic engineering method that uses molecular scissors or artificially engineered nucleus to target and induce the double-stranded DNA break at a specific location. So, these are the molecular scissors or the genome editing tools, which like a zinc finger nuclease, transcription activator like effector nuclease, homing endonuclease, and CRISPR-Cas9, that is clustered regularly interspace short palindromic repeat. So these genome editing tools, what it do? It create a double-stranded break in the genome, and this double-stranded break, which is further repaired by the either non-homologous end joining mechanism or homology directed repair mechanism. And while the repairing, there is a mutation. So their mutation takes place here, and that is not, and this mutation can be insertion, deletion, or addition of the specific desired fragments. If now we'll see the popular technique of this genome editing, that is the CRISPR-Cas9 genome editing. So when it was emerged, so the CRISPR sequences were first discovered in 1987 by Shu and co-worker, and further these Cas genes were identified in the prokaryote, which are associated with the DNA repeats by Jensen and co-worker. And in 2007, Bangalore and his co-worker, they have identified that the CRISPR provide acquired resistance against viruses in the prokaryotes. And the idea of the CRISPR-Cas9 as a genome engineering tool was published first time by in 2012 by Jennifer Doudna and Emmanuel Charpentier, and they received the Nobel Prize for this specifically use of CRISPR-Cas9 as a genome engineering tool in 2020. So Nobel Prize in chemistry was awarded for this discovery. And since 2013, this CRISPR is used in all eukaryotes like human, mouse, plant cell using the design CRISPR RNA sequences for genome modification. And later on, you can see the history here. You can see here in 2013, 98 articles are published in PubMed on CRISPR-Cas9, and later on the graph is increasing. So this is how this popularity and use of this particular technique in the life science. Now we can see the mechanism of this CRISPR-Cas9. So basically this CRISPR-Cas9 is adaptive immune system in bacteria. So each CRISPR locus is flanked by the AT-rich leader sequences and followed by a series of direct repeats. So separated by unique spacer sequences acquired from invading genetic elements. So these are the CRISPR locus. These are the genes which are located in bacterial genome, and this bacterial genome, so where it has a clustered regularly interspaced short palindromic repeats. So these are the repeats, and these are repeats are in between there are spacer sequences. And what are these spacer sequences? The spacer sequences is nothing but the memory of bacteriophage or foreign DNA which is infecting into the bacteria. So from these bacteria, these spacer sequences, unique sequences which is having adjacent protospacer adjustment motif, they are acquired in this CRISPR locus. And when next time the similar kind of bacteriophage infect into the bacteria, so this gene will be activated and it will provide the CRISPR RNA, and this CRISPR RNA find the complementary protospacer which is already there, present there in the this genome of this bacteria, and it will be identified by the this CRISPR RNA associated with the transacting RNA. And to this the Cas9 protein recognize this particular, and this Cas9 protein create a double stranded break here, and this break is further repaired. So this protospacer, they are flanked by a short motif called as this protospacer adjustment motif. So this protospacer adjustment motif is very important. And the long CRISPR transcript, that is pre-CRISPR RNA, are processed into the short CRISPR RNA that guide the Cas9 protein to invading nucleic acid through complementary base pairing. In this way, this is how this particular CRISPR-Cas9 mechanism present in the bacteria. Now, how this mechanism is used for the genome editing using CRISPR-Cas9? So, if we here, if the idea is that if we want to create a mutation in desired or the specific gene, what we can do? We design the CRISPR RNA sequence. So, this is about 20 base pair target specific guide sequence. So, we design this sequence, and we put this Cas9 protein along this sequence, that is CRISPR RNA sequence, along with the Cas9 protein. So when it find the specific target in the genome, what it will do? It will create a Cas9 mediated DNA cleavage double stranded break, and this double stranded break is repaired by the non-homologous end joining or homology directed repair mechanism. So in homology directed repair mechanism, we can use artificial repair template which is inserted here while by the mechanism of homology directed repair. And in non-homologous end joining, this double stranded break is repaired, and where while repairing there is a mutation, that is either small insertion or deletion, and this mutation alter the structure of the gene, and finally the gene is edited. So when we see how this CRISPR-Cas9 is used in the genome editing for plants, so there are the three steps. In first step, first of all, we have to select genomic target and design single guide RNA about 20 base pairs. So target gene will be identified, and based on the sequence of the target gene, design a 20 base pair guide RNA sequence which is having single guide RNA backbone, and which is put under the promoter U6 or U3 promoter, and further this will be the expressed as a RNA sequence. In the second step, combine this, assemble the single guide RNA and Cas9 construct into the suitable vector. So here you can see your target sequence, guide RNA scaffold, and here U6 promoter which is put under the promoter of RNA promoter, and here you have a CMU promoter under the Cas9. So Cas9 protein is put under the CMU promoter, and Here you can see the linearized construct, single guide RNA Cas9 construct for plant transformation. So when it is expressed, you get the RNA for guide RNA and the protein of the Cas9 protein, and they both will work together for creating the double stranded break. Now, in third step, what we'll do, we will validate the single guide RNA by the transgene expression in the protoplast. So here, protoplast, what is protoplast? Protoplast is the cell without the cell wall. So we extract and transform protoplast with the plasmid carrying Cas9 and single guide RNA. And after the treating it, then extract the genomic DNA of the transformed protoplast of Cas9 and single guide RNA and check whether the mutation is taken place in the desired gene, target gene or not. So it can be checked by the PCR amplification or restriction digestion or sequencing method. So these are the different methods used for the checking. So how we can, once we found that okay, our construct, that is our single guide RNA and Cas9 is working fine, then we can use this Cas9 and single guide RNA for delivering into the plant cell and tissue, and several methods are used for the delivering this Cas9 and guide RNA into the plants. For example, floral dip method, agroinfiltration, viral vector method, particle bombardment, which is direct method of plant transformation. Then we can also spray the guide RNA solution onto the plant where we express the constitutive expression of the Cas9 protein. And another important technique, that is DNA free genome editing, where we are not inserting any foreign DNA into the plant. We are directly using the ribonucleoprotein, only protein and the single guide RNA, and that will be delivered into the cell, plant cell, where we didn't have any foreign DNA. So that protein and that guide RNA will do their work, and there will be no any evidence of the this DNA. So that DNA free genome editing methods are nowadays getting very popular. Now, once you have delivered the plant, when we get a transgenic plant, then regeneration and screen the transgenic plant for the genome editing event, so that we amplify the specific gene using the gene specific primer, using the technique, that is a PCR technique, and it will be checked by the high throughput DNA sequencing or restriction enzyme site loss or the surveyor assay. So you can identify whether their mutation is there or not. You can see here mutagenized and digested plant, and here control digested. So if there is a mutation, we will not get the desired bands. So now we will see how this CRISPR-Cas9 technique is used for the crop improvement. So for disease and pest resistance, for example, rice blast resistance can be achieved by knocking out the ERF transcription factor gene, that is OSERF92 in Japonica rice variety Q131. Further, the canker susceptibility gene, that is a CSLOB1, was modified using CRISPR-Cas9, and plant were resistant to the citrus canker. And then quality improvement was also achieved by using the CRISPR-Cas9 technique. For example, low phytic acid in maize, then the fatty acid composition is enhanced in the oilseed crop, for example, camellia sativa and other allohexaploids, and in abiotic stress tolerance, for example, mutation in gene, open stomata gene, which is a major plasma membrane ATPase in stomata response in Arabidopsis. So by mutating such a gene, the plants for resistant to abiotic stress was achieved. Here you can we can see with the example. See here the starch in potato, when stored into the cold storage, the enzyme, that is vacuolar invertase, converted into the glucose and fructose, and when we use such potatoes, cold store potatoes for preparing the chips, then it will get the toxic compound, that is acrylamide, and dark brown pigment is formed. So to prevent this, these vacuolar invertase uh, gene enzyme encoding the gene of this vacuolar invertase is mutated, and the potato form that are innate potato. So this is the commercialized technique. Then also polyphenol oxidase gene was mutated using CRISPR-Cas9 technique in the apple, and the browning in the apple after cutting is inhibited, is prevented, and the apples are called popularly called as Arctic apple. So when we cut apple and we get exposed into the this is specifically useful in apple juice, so that will not be prevented from the blackening. Similarly, caffeine production is also reduced using the mutating the xanthosine methyl transferase gene in coffee. So let us see what are the these are the some few commercial example, but there are many more. So now we'll see what are the biosafety regulations associated with this CRISPR edited crop. So current US regulation has already classified a number of crop mutated by the synthetic nuclease as a non-GMO, and they contain an induced mutation but no transgene. And the offspring of the transgenic plant parents, which themselves lack a transgene, would not fall under USDA's regulatory authority. Then US Department of Agriculture allowed the cultivation and sale of CRISPR edited mushroom without passing any regulation. Further, the plants where mutation are induced by proteins such as synthetic nuclease or ribonucleoprotein, that's what I have explained, DNA free genome editing. So these are not fall under the current European Union regulation and qualify the respective plant as a non-GMO plant, and several countries have already formed a regulatory framework for genome editing technologies like product-based and the process-based framework. So here we can see the USA and Canada, they have obtained the product-based regulations. Asian countries are still framing their regulatory for this CRISPR genome editing crops. And first, the South America, which is the first country to pass the genome editing technique, that is the NBT technique. And so now we'll conclude our topic. So if we see the CRISPR technique, so it is the CRISPR technique have ability to generate a highly specific, rapid, cost-effective, and efficient mutation in short time span. And this CRISPR technique give a sustainable solution for global food crisis, like CRISPR-Cas9 technology show great promise for speeding up both forward and reverse genetics, and will enhance the basic plant biology research field. But still, it is a challenge to completely reduce the off-targeting as well as the accurate detection of off-targeting in a whole genome. There is also need of a single guide RNA libraries for genome-wide detection of the multiple mutations in a single experiment like animal system, and there is a need of harmony at international level for regulation of genome editing crops to assess the potential risk associated with them. The crop genome editing will bring about the next green revolution. So this is a very promising technique and will bring a next green revolution. These are the some references which I have utilized for preparing this presentation. If you have any questions, please ask. Thank you.